Hello and welcome back to the Computational Analysis Lab. Today we are going to be talking about transcripts per million. Now you'll see this a lot in RNAseq data and you're starting to get some really good skills. You've got all your significant genes and that we did everything up into this point using the counts matrix from our NF core RNAseq pipeline. But there was another file that came out of that pipeline that had TPM values in it, salmon merged gene TPM values. Both of these files are in the shorts on the channel so people know what data files to have. But really those two files, the counts table and the TPM files coming out of the RNAseq pipeline are the two main things we need. So today we're going to work on that other file, the transcripts per million. Now this is kind of complicated, but I'm going to make it fairly simple, but there's a lot more explanation here in the R Markdown file. Oh, again, we are in 06 and we're doing 05 TPM transcripts per million. I'm going to set my working directory as usual. And so we can just do read.table. It's in your NF core RNAseq output star salmon folder, and it's salmon.merge.gene_TPM. And there's a lot of complicated nuances here, but really what's happened is salmon started all the alignments to the genome, and salmon likes to count them. And so we had that counts file from salmon, and also a TPM, which says how many reads were there for this gene per millions of read that were run on a sequencer. And also that's important because if one of your sequencing samples had less reads, you want to have it normalized to a depth of sequencing reads. Now you might have heard of RPKFMs, reads per kilobase per fragments per million. It's the same kind of idea. In that case, you're taking the gene, you're counting salmon's saying how many counts did I get across this gene. I'm going to divide by the length of the gene because the longer the gene I'd probably get more counts just by chance, and then how many millions of reads were in there. Very simply, you can just think of it as an abundance measure like we did with base mean or counts. And different plots are going to use different measurements. I try to use counts as much as possible because there's more measurements there. And again, another caveat with TPM or transcripts per million is that it's giving you one value across an entire gene. Okay, so let's read that in. Yours will be wherever you had your pipeline output for our mouse doc-seq. Okay, so we see the same number of genes, which is good. We had it for our counts table. And let's just take a look at that. And so we see it looks very similar. No gene ID or gene name column. That's nice. Thanks, Salmon. We have our sample names up here in meta, and our gene ID also here in meta, and then our values there. So you would never put TPMs into DE-seq. I'll say that now. You would find out pretty quick. If you had done that, always use counts for DE-seq. TPMs are more for plotting, which is where we're moving towards, is making some pretty plots as a measurement of how much the gene there was across time and how much it was changing. You can also use log fold change, which is going to be based on the base mean from DE-seq. I prefer that again, but I just want to make sure everybody's on the same page with what's out there. Okay, so first thing is a lot of our TPMs are going to be 0, which means the gene just wasn't expressed in that cell. That's typical. Usually more than half of the genes are not expressed. And so we're going to make TPM 0. So we're going to make a data frame or matrix of just all the genes that are 0 in every sample, because we're probably not going to want to use those genes. Okay, so we're going to say go into this TPM object we just read in, square bracket index, and we're going to do row sums as a function inside that indexing. So this is the row portion of the square bracket indexing, and we're going to retrieve all the columns for all of our time points. And so we're going to say if the row sums of TPM is directly equal to 0 across all the columns of TPM, keep those rows. And you'll see why in a minute. And keep all the columns. Okay, so let's do that. And so now when we look at this, we should just see a matrix of zeros. Should be loading here. And we can see it's about half our genes, again, 23,000, and it's going to be zeros across all the different time points. I'm not going to scroll because my browser will change back, but okay, so that was successful. And so now we could subtract those out. But for now, we're going to take, oh yeah, we're going to do that, but our gene ID is currently in the black section or in meta. We're going to do row names to column here, and we're going to keep the same name. Always be a little careful of that, but we're going to take this TPM object and we're going to move the row names to a column, and that column is now going to be called gene ID as we've been using it. And we'll see that we now have those gene IDs moved in as actual values in the table. I don't know why this is taking so long. Sometimes it's faster if you just click over here. And we don't see that. Oh, that's because we didn't do this TPM. And so we see now our gene ID is in the columns. Okay, so we are going to do the same thing to our TPM 0, and we'll just subtract them out. It's kind of around the elbow. There's other ways to do this. You could unfilter by row sums equals 0. Just trying to show different ways to do to learn more R code. Okay, so we're going to do the same thing for our TPM 0. So now we have a column in which the two tables match identically, or that we could use to subtract them. Okay, this is going to look funky, but I think the main lesson here is that when you square bracket index into a table, you can do so much in there, and just have rows separated by columns with the comma. But here we're going to see our first not sign. So exclamation means not. So you always read sort of left to right, but we're going to say in my TPM gene ID column, in is a nice tidyverse function. So it's saying if my TPM gene ID is in TPM 0, don't take it. That's the not sign. So it's almost like psych or not. It's saying TPM gene ID, if it's in the TPM 0, don't take it. So again, there's lots of ways to do the same thing. Just thought I would show that and also highlight this in function as kind of saying if it's in this, then keep it or don't keep it, and something we'll use quite often. Okay, so now TPM not 0, so our TPM is 55,000, and it should end up being about 22,000 when we subtract these two numbers. Okay, let's take a look. And that's about right. So these two would add up to this number here. And we can see now that in this TPM not 0, we're going to have values in every single spot, and there will be no row that equals 0. Okay, great. So now we know how to get rid of our genes that don't have any values in them. And so let's go look at them. So we're going to use this TPM not 0. So we filtered out anything that is 0 across all rows. And let's do a histogram of just the first column, the wild type 0 time point replicate 1, setting the X limits to 0 to 300. I already did this, so that's why. And 1,000 breaks as we did in the previous class. Okay, let's take a look at that. Nice. So we see again the same thing we saw with base mean, that most of the things are here and then they spread out really far. So most of the things look like they're between 0 and 20. Let's take more look at this here. So we're going to move our row names back in. Okay, so this is just to show more R code. So if we look at our TPM here, this might be faster, double click it. We can see our gene ID is in there. Let's take it out so we can start to do some analyses across this table without having character values in it. So we're going to say the row names of TPM is TPM gene ID. So that gene ID column is still going to be there, but we're going to now make a row names for that. Okay, let's look at that now. So now we have double here. I think this exercise is just to show you how, if you're going to do some analyses, you want just integers in your table or matrix, you want to move these to that row names. And then we can remove that gene ID column. You've seen other ways of doing this. It's just another one by saying null. Just remove it, that it has no value to us anymore. So let's look here at TPM, no more gene ID column. It's just another way to get rid of a column. Okay, and then we are gonna take our columns to row names for TPM and call it gene ID. So we're moving it back in. This is just kind of learning some basic R. You saw right now that the gene IDs are in the meta. Let's just skip that and go to row sums. So now we're going to take our TPM. We're going back to our original one. This is a common thing is in RNAseq that you want TPM greater than 1. Moving forward, we're not going to do that, but it's a good way to learn some filtering. So we can just go into our TPM, take row sums greater than 1. So that means that at least one value in that row was greater than 1, whereas previously we just removed all the zeros and keep all the columns. Okay. And now let's make sure that any row sum of our new object is less than 1. This should be false because we said, yep, so the answer is false because we filtered on things being greater than 1. And as a positive control, we can say, are any of the rows in the TPM less than 1? And the answer is true. Right, yes, because we filtered greater than 1. Okay, so yes, so this is all making sense, and now again we can plot that again so we don't have so many zeros in there. With this hist, we're going to TPM, first column is that, setting the X limit to between 0 and 30 and 1,000 breaks, and we zoom in. It looks pretty similar, but we can see a little more fine grain granularity here, that most of our TPMs are here, and then many of them are not. Most of them are not greater than 50. So it's just good to know when somebody says TPM of 1, that means that actually most of your data is a TPM of 1. So, okay. So we've done a lot of good filtering. So what we could do is take a mean TPM. So we'll take the mean of the first column of TPM and just find out what is the average TPM across a given RNA-seq sample. So this is just our one sample, 0 time point, replicate 1, no dox exposure. What's the mean TPM? So if we look at that, it's about 45. So on average, they're about 45. But if we look in our non-filtered, so this is when we filtered out those low rows that added up to low TPMs. If we look at the non-filtered ones, the average is 31. So when we filtered those out, they tended to be higher. This all should sort of make sense. We're just getting a feel for TPMs here. And so let's take the average across the rows. So that's pretty handy. So you can say, what is the average across all the rows? Like previously we did row sums to get to 0. Instead now we're going to do row means. We're still going to go through every row and say like what's the mean value there. And so we can look at row means here. And maybe we shouldn't because it's quite large. But it'll give you, now we can say what is the mean across all time points for any gene. So we just did, we took our TPM file and we did row means. We said what's the mean for one row, which is a gene, and it did that for all the rows, and it kept the mean value in this object down here. And then now we're going to say what is the average of that. So what that would mean is what is the average TPM for all genes is essentially what that will get us to. Okay, so we made this object row means, which is the mean TPM for every gene measured in TPM filtered, and we can find the mean of that, and it's 45, which is the same thing we saw above. So basically, if you just take a given column, you get the same answer as if you take the average across all the columns. Okay, so you probably know too much about TPMs at this point. If you're, just trust me, this will all make more sense as we start using the practical things. But I just want you to have a place to come back and start to look at that. So now let's look at the standard deviation of the mean. So we see that a mean for one sample is 45 in our filtered TPMs. And we see that if we average across all time points for all genes, we get the same answer. But look at the standard deviation, which is this SD function here, 310. So that means that the mean is 45, but the standard deviation is 310. That's a lot. So that means there's a lot of variation in here. Just to say, be careful with any value. Don't be wed to any single measurement of abundance. The variance is very high for TPM. Okay, so let's look at how many genes have a TPM of average of less than 1. So if we say what's the average across all the samples, and if we did that for all genes, the average of that would be 45. But now let's take how many, we'll do this row means on TPM filtered again, and then we're going to index into that row means as it's thinking about it, and we're going to do the row means again for all the values that are less than 1. So that's going to say that in this row, the average is 1 for the TPM. So that means what you'll hear a lot is, oh, TPM greater than 1. And I think this is why I'm not so sure about that subjective threshold that has come to be. So if we look, and then length will tell us how many observations there were. So row means, so we're going to go into the row means function of TPM filtered, where it's taking the mean across every row, and it's keeping each mean for every row there is, so 22,000 rows, but one value. And then we're going to say, tell me the mean of that of less than 1. Here, 8,333. So roughly a third of our genes are less than 1 TPM. So just to say, kind of be careful when people say we require TPM of greater than 1. And so there's some more things to think about in here and what we just did. But what we really want to do is find out what's the average and standard deviation, because we have 3 replicates for each time point. So what is the average TPM for time point 0, and what is the standard deviation for time point 0? So that's going to need some more data manipulation here. And so we're going to make an object that is simply our time points. So we're going to call it time points. And C just means like paste this, and again with the quotes, 0, 12, 24, those are our time points. And we have replicates in our TPM filtered here that are underscore R1, R2, R3. So we can use that as a handy way of knowing what the replicate number is. So we could say 0, R1, 0, R2, 0, R3, and get all of our data back. Okay, trust me, there's a reason for making this. Okay, so now we're going to do another for loop here. And so we're going to find the average for each time point using a for loop. So we're going to say for time point. So you could use I here, you can use X, you can use your name, whatever it is, it doesn't matter. It's just what the computer is going to be thinking about for each iteration of the loop. So we'll just call it TP in time points. So we're going to have 5 time points that we're going to loop through. And so for we're going to make an object called columns or calls, and we are going to grep anything. So grep is, we did this in bash, we'll grab anything that matches the string, and we're going to make the string for it to match using our time points and replicates. Okay, so every sample has wild type, so grab that, combine that with the time point you are on. So the first one would be 0, the next after that will be 12, 24, 48, and then underscore R, that will get us all of the columns to be able to grep them out, and then make those the call names of TPM. Grab that string. So this is the grep string. We're going to get wild type underscore time point, that it's in the loop, and underscore R, that should match all of our data, in the call names of TPM filtered, which has all of our sample names right here. So grep is going to come into this column names and just find anything that matches. So in the first iteration, it's just going to grab these 3 columns. So it's going to say in my first version, I'm going to look for 0, and I only see 3 zeros. I'm going to keep these 3 samples, and then we can do a mean and a standard deviation for those 3 things. So grep is doing a lot of the work to just grab the time point, and then we can do some calculations to it, which is average. So now in this first one, for each of the 3 replicates, it's going to take the average for all the genes in the table. Okay? So that is where we're going to use the average, get the average value, which is the row means of TPM filtered. So we're going into the full data set there, and take all the rows, so all the genes, and only give me the columns that have matched that grep. So it's only going to be time point 0 the first time with the 3 replicates, etc., and then the different time points each other time. And it's going to take the mean and give us, so basically, I shouldn't have put average here, it should be mean, but I think you get the idea. We have 3 replicates, and we want to have 1 TPM value for those 3 replicates. Okay. So then we can add this to our list as we're going. Oh, sorry, we also made a generic list here. So X average value is a list, and now this for loop is going to populate that list. So X average values, and we're going to then take this, our average, and so we're going to grep into the columns and get the average here, and we're going to concatenate this X average values, which is just a list right now, and list the average for all the rows. Okay, I know this is a bit confusing, but go back through it and you can see, do some more, do it a little slower, and that's going to end our for loop there. So we're going to have lists that have the average for all the different time points, and then we're going to use do calls, base R saying do this, essentially. We're going to column bind. So now we have an average for 0, 12, 14, 24, 48, 96. We're going to column bind. Before we row bound, we bound all the rows together. Now we're going to say I have a list for 0, 12. I'm going to merge all those into one table with this cbind function. And what I'm going to bind is the X average values that were produced in this list here. Okay, so let's do that. Let's run this for loop here. And so what we can see in our X average values here is a large list. Again, it's in this weird format that's not so handy, and that's why we're going to do this column the column binding in a second. But we have 5 time points, and now we have an average value across the 5 time points. And so let's do this column bind on that object we just looked at. And so it went in there and it found all the columns, and it gives them these weird names, V1, V2, V3, V4, V5, because those were the lists. We could have named them, but we can name them right now. So we can say the call names of this X average value list we just made here, that's starting to look like a table, is the same as the time points. Okay, so do that, and now if we look at it, we have 0, 12, 24, 48, 96 for our time points. And then we can convert this back to a data frame as .dataframe, X average values, and this will give us our average TPMs here. Okay. And then now if we look at this, we have a nice handy table. Average TPM. Why is it taking so long to load? There it is. Okay, so we have our meta gene IDs and our meta time points, and now we have a bunch of data that we could use for analysis. But again, all that was to do is say we've got 3 replicates, I want an average for each one, and now we can do the same thing and get the standard deviation. We need to install this package, matrix stats. So I'm not going to install it, I already have it installed, I'm just going to load it. Same thing. So we're just going to reiterate through this same kind of for loop logic that we just did. We're going to make our time points and replicates so we can grab them in our grep. And then we are going to make a list, but now of standard deviations. So average and standard deviation. I think we're going to do both of them in this for loop. So that was just to get, because I know it's really confusing getting through these for loops. Trust me, at least I was confused. But now this might make more sense. We're going to do both the average and the standard deviation in the same for loop. And we needed that package because it does a row standard deviation. Believe it or not, there's row means, but there's no row standard deviations without this other package. Okay, so let's make a generic list. So each time the for loop goes, it's going to make one of those weird looking data frames where with the 1 and the 2 square brackets, it's going to say this is what I got for this list, each time it goes through the for loop. And it'll do that 5 times, once for each time point. Okay, so we just made a generic list. There's nothing in it. And so same sort of structure for time point in all the time points, which is what we just made here, 5 of them. To get the columns, go grep the wild type because it's on there. To start with the time point we are currently doing, which is 0, and underscore R, so it'll get all 3 replicates. And then it's going to take and look for that grep function in the call names of TPM. So this grep needs, what am I looking for? Let's paste 0 here. And then where do I look for it? And that's where we're going to look. Then we can get the average through the row means. So in the first iteration, we're going to have the 0. So we'll get the average for all the genes. And then the same thing is we will do for standard deviation. I'm not sure if this will work with apply, but basically it's going to go into TPM filtered and get all the rows and only the calls that are in the for loop currently and calculate the standard deviation. And that's from this package that will give us the row standard deviation. Okay, so then we're going to make a data frame for standard deviation, and we have one for average already, that list, and then we're going to merge the 2 in there. So again, I appreciate that for loops are tricky. You could also do this whole thing with lapply, but let's give it a run here. Okay, so, and then let's do that same converting to a data frame. So we're gonna, well, let's look at it first. Okay, so we have this output of average and standard deviation values, average and standard deviation. So we got a list because we made a list at the beginning, and we have the average and the standard deviation. That's why there's now two columns. So there's 21,000 rows for each gene in our filtered TPM file, and 2 columns, one with average and one with standard deviation. So then we got to pull that out by column binding within each of those lists. And we're going to do that in this average and standard deviation values object we just looked at. So now it's going to look more like a table. There we go. So we have the average and standard deviation. The next one. It automatically named these columns. So we're going to want to rename those columns, where we're going to say the call names of that object is going to be the replicate and time point. And there's 2 things, average and standard deviation. Okay, so we look at this now. Let's take a look at our final product. And then we are done. I know this was a lot. I think it's the most important thing is going in and saying, if I have replicates, how do I get an average and a standard deviation for them? because oftentimes you're just going to want to make a bar plot of the 0, 12, 24, 48, 96. And so now we can see we have the 0 average, 0 standard deviation, 12 average, 12 standard deviation, and so on. Awesome. We are ready to start plotting TPMs, but I have an exercise for you to go through. I'd go through the for loops a few more times, and you can go row by row if you want, and get a feel for those, but that's something you're going to want to do a lot of, is saying I have multiple replicates, but I need to get the average, and always keep the standard deviation as you're doing that. And so this has been a lot, but you have now learned almost all the basic fundamental analyses you will need for RNA-seq. In the next class, we're going to put that all together and see actually how simple it can be when you don't have to learn each step. We'll make a streamlined RNA-seq pipeline. So see you there.